Nobel laureates, excellencies, ladies and gentlemen, a very good afternoon to you all. It's a great pleasure and a privilege for me to welcome you all to Karolinska Institutet, its Aula Medica building, and the 2015 Nobel Lectures in Physiology or Medicine. My name is Anders Hamsten. I serve as Vice Chancellor and President of Karolinska Institutet. The core of the Nobel Week, a particular highlight of which are the Nobel Lectures, is to celebrate science and to rejoice at the unique contributions of some exceptional individuals. Research is a way of slaking our curiosity about life, testing our own limits, and having an outlet for our creativity and competitive spirit. In the spaces of physiology or medicine, the ultimate goal is to achieve scientific breakthroughs that change our view of human health, disease, and normal vital processes. It is an honor for me to extend my warmest welcome on behalf of Karolinska Institutet to Satoshi Ōmura, William Campbell, and Tu Youyou, the 2015 Nobel laureates in physiology or medicine, who received the Nobel Prize for their discoveries concerning a novel therapy against infections caused by roundworms, parasites, respectively discoveries concerning a novel therapy against malaria. Without further ado, I now call upon Jan Andersson, Professor of Infectious Diseases and member of the Nobel Committee, who will introduce the laureates.
Dear laureates, distinguished guests, friends and colleagues, I'm privileged and honored to introduce the three Nobel laureates in physiology or medicine 2015. Dr. Satoshi Ōmura, William C. Campbell, and Tu Youyou. Together, these three scientists have fundamentally changed our ability to treat parasitic diseases, in particular roundworm infections and malaria. Parasitic infections afflict more than half of the world's population and impose a substantial health burden, particularly in resource-poor nations. More than 1 billion persons worldwide are infected with intestinal nematodes, and more than 3.4 billion people are at risk of being afflicted by malaria. The discoveries of the laureates have led to unmatched advances in human health. Finding ways to understand and treat infectious diseases has a long Nobel history. Charles Leveron, Ronald Ross provided important insights into the cause and transmission of malaria. In the area of bacterial infections, Gerard Domack was awarded for sulfonamides, Alexander Fleming, Ernst Chain, and Howard Florey for penicillin, and Selman Waksman for streptomycin. Gertrude Elgen pioneered the field of antiviral treatment. However, this year is the first time a Nobel Prize in physiology or medicine is awarded for the treatment of parasitic infections. Satoshi Ōmura initiated already in 1965 attempts to identify agents with antimicrobial activities from a group of bacteria, Streptomyces, which live in the soil. He developed extraordinary skills in selecting unique bacterial strains and how to characterize them. Indeed, from more than 40,000 different Streptomyces cultures, he was able to identify a completely unknown type, Streptomyces avemactylis. That contained a compound never seen before and which would change the history of medicine. William C. Campbell received the Streptomyces strain from Omura and identified the unique properties of Streptomyces ivermectilis in killing many different intestinal nematodes with extraordinary potency. The effect was exerted by ivermectin, which was modified to the more effective compound ivermectin, with a single-dose activity lasting for more than 6 months. Ivermectin was well tolerated, killed both roundworms and their eggs in various host species, and was effective also against nematodes resistant against existing drugs. Inspired by these results, Campbell initiated successful trials in humans with nematode infections causing river blindness or lymphatic filariasis. Tu Youyou had a strong vision that herbs may produce potent compounds against malaria parasites. She consulted ancient Chinese literature on herbal medicine and found evidence that extracts from many different species had been used to treat fever. In order to assess if this was associated with potential anti-malaria effect, she used extracts from more than 200 different herbal plants. The plant Artemisia annua, or sweet wormwood, emerged as an interesting candidate. But the results were inconsistent when used in experimental malaria model. Based on suspicion that the active component in the Artemisia might be sensitive to the extraction process, she improved the extraction technique to achieve better purification, eliminated a toxic contamination, and now demonstrated a potent and consistent effect in mice and in non-human primate models. Not only did the extract reduce the fever, but it also rapidly eliminated the parasites. The isolated active component in the extract was named artemisinin. By establishing its chemical structure, it became clear it represented a new class of anti-malaria agents affecting the malaria parasite at an early phase of replication. This explained its unprecedented potency in the treatment of severe malaria. The discoveries of Omura, Campbell and Tu are of immense importance to treat parasitic diseases, primarily affecting the world's most vulnerable citizens, particularly in sub-Saharan Africa, South Asia, and South and Central America. Roundworm infections often occur during childhood and give rise to lifelong suffering and disability. Currently, 25 million inhabitants in 37 different countries are affected by river blindness. 800,000 people have impaired vision or are already blind. More than 120 million people suffer from lymphatic filariasis, a disease leading to swelling of limbs and other body parts, immobilization and stigmatization. Given the efficacy of ivermectin, global initiatives sponsored by WHO and NGOs have the goal now to eliminating these two diseases by 2025. More than 200 million cases of malaria are reported yearly, leading to the death of 500,000 people, predominantly children below the age of 5. Artemisinin is used as a combination therapy in all malaria-ridden parts of the world and is estimated to reduce mortality from malaria by more than 20% overall and by more than 30% in children. For Africa alone, this means that more than 100,000 lives are saved each year. Therefore, the WHO guidelines are now recommending that every patient with falciparum malaria should be treated with artemisinin-based combination therapy. So in sum, the discoveries from this year's 3 Nobel laureates have led to immeasurable improvements in human health and well-being. And we will now hear the fascinating stories of the discoveries from the laureates themselves. So let me therefore, without further ado, introduce our first speaker, Satoshi Ōmura. Satoshi Ōmura received a PhD in pharmaceutical sciences in 1968 from University of Tokyo, Japan, and a PhD in chemistry in 1970 from Tokyo University of Science. He initiated his research at the Kitasato Institute, Tokyo, Japan, in 1965. He moved in 1971 as a postdoctoral fellow to Wesleyan University. Subsequently, he became professor at Kitasato University in 1975, and Omura is currently professor emeritus at Kitasato University. Please join me in welcoming Dr. Omura, who will present his Nobel lecture under the title, A Splendid Gift from the Earth, the Origin and Impact of Ivermectin. Please, Dr. Omura.
Thank you for that extremely kind introduction. It is a very great personal pleasure for me to be in this wonderful forum today and to be able to share with you some of the joy and the result of my research. Firstly, I'd like to thank the Nobel Committee and all of those responsible for identifying my work as being of both major importance and significant benefit to mankind, and worthy of Nobel Prize. It gives me the greatest pleasure and deepest honor to share this prestigious award. And I do so in the knowledge that I am here representing a large and diverse group of individuals and agencies, all who deserve to share in the recognition of unmatched success story in human health. Ivermectin appeared through a collaboration between the Merck pharmaceutical company and my research group. And to start with, I would like to explain a little about our work at Kitasato. My research on microbial metabolites started in 1965 at the Kitasato Institute. I began with the structural analysis of the clinically used antibiotics, leucomycin, and other antibiotics discovered at the Institute. Soon afterward, I decided to focus on research for novel, natural occurring substances which possess biological activity, particularly those produced by microorganisms. Ever since, I have been involved with devising new methods to isolate microbes from samples from nature, culturing them, and creating and exploiting novel, highly specific screening systems to detect interesting chemicals. These are the steps in our research. We isolated more than 2,000 microbes each year from among the hundreds of thousands that exist in the small sample. We then cultivate and grow them on different media. They are then put through a variety of screening systems to identify any interesting bioactivity. Those producing promising biochemicals are identified and preserved for use by us or by other scientists. Promising chemical or microorganisms go on for further research and development. This, of course, is expensive, and R&D usually requires a commercial partner. This was the initial pathway that eventually led to the appearance of ivermectin. This is an example of diversity of microorganisms that have been isolated from soil as opposed to the root, plant root. There is a distinct, divergent, and extensive variety between the two. We have been able to discover many new organisms as well as chemicals with interesting bioactivity. you can begin to visualize the as yet undiscovered potential that microbe possesses. Our research has resulted in the discovery of an average of 10 novel compounds every year for a period of almost 50 years. Among them, 26 compounds have been widely used as medicine in human and animal health and as biological reagent. Among the compounds we discovered, over 100 have become the target for the synthesis in organic chemistry, meaning that our compounds have also contributed substantially to worldwide advancement in organic chemistry and biochemistry, including making a contribution to other Nobel Prize winning research. This is an overview of microbial metabolites we have discovered so far. Now, I would like to concentrate on the ivermectin and ivermectin story. Fortunately, in the early 1970s, Professor Mike Tischler of Wesleyan University, former ACS president and former president of Merck Sharp and Dohme Research Laboratories, introduced me to Merck. This allowed me to start a novel international collaboration with Merck company. It represented the world's first major public sector, private sector partnerships, a term which I'm sure that most of you are now familiar with. Professor Tischler invited me to work with him at Wesleyan University in 1971, and we became the close friends. He played a central and key part in setting up the collaboration with Merck. At this point, I have emphasized that scientific research cannot be done in isolation. It is not an individual endeavor. I have been able to work with some wonderful scientists and colleagues from many disciplines and from many countries. Ivermectin originated in Japanese soil, but it would not have seen that light of the day were it not for the excellent teams of scientists at Merck Company. You see some of the key personnel involved here, and I would like to draw attention to the very special role played Merck's. Today, scientific progress is increasingly being achieved by a group of multidisciplinary scientists. Ivermectin was a pathfinder in this respect. As you can see from this selected sample of product, our joint research discovered many biologically and structurally interesting metabolites. For example, the ruminamycin and rastromycin are antimicrobial, active against anaerobic bacteria. Elastinin is the first human elastase inhibitor of microbial origin. Cetamycin is a VATP inhibitor, and this compound is very important biochemical reagent, and alpha-chain renin inhibitor. All of the metabolites we found, ivermectin, co-discovered with William Campbell's teams, was the most unique and the most important. It was the world's first insecticide, a completely new class of drug, capable of killing organisms inside and outside the body. Ivermectin was changed by Merck's chemistry group into a safer, more effective compound, a dihydroderivative, ivermectin. Among the first 50 promising organisms that I sent to Merck for testing, this one had the most potent shell. It produced several interesting metabolites. Campbell's teams found that it had good anthelmintic activity. Based on our genetic study, we named it Streptomyces aberamytilis. It was later renamed Streptomyces aberamyctineus. Despite decades of searching around the world, the single organism we discovered in Japanese soil remain the only source for industrial production of ivermectin ever found. Ivermectin blocks nerve impulse transmission in glutamate-gated anion channel, causing paralysis and death in parasites. In 1981, ivermectin-containing products were commercialized for animal health, and it proved extremely safe and effective for human use. Merck scientists, working with a collaboration with the World Health Organization and disease-affected communities in Africa, soon discovered that ivermectin was the ideal tool to tackle a disease that had plagued tropical regions for centuries. Onchocerciasis is also known as river blindness. It causes skin disease, blindness, and sometimes death. It also prevents socio-economic development in all endemic areas. In the late 1980s, well over 120 million most poor people in Africa and Latin America had no access to a suitable solution to the disease. No safe, effective drug was available until ivermectin appeared. Massive clinical trials with ivermectin were held in tropical Africa by WHO, TDR, and other organizations. They showed that a single annual dose in ivermectin, 200 micrograms per kilogram of body weight, eradicated microflare worms from the eye and skin after 1 month, and that the patient remained free of immature worms for up to 12 months. Ivermectin was registered for human use in 1987. Merck, under the direction of Dr. Roy Vagelos, immediately started a unique donation programs. Under the brand name Mekuchizan, ivermectin was to be proved free for the treatment of river blindness in humans with the Kitasato Institute for Going Royalty. The donation was for as much drug as needed, for as long as needed. This created the world's largest, longest running, and most successful drug donation programs. Using this Mekuchizan tablet, a formulation of ivermectin for human use, continued community-directed mass treatment with villagers themselves. Distributing tablet is now used throughout Africa and in other endemic countries in Latin America and in Yemen. Donation does not mean that it reaches the people who most need it, especially if they are in the poorest and most remote community around the world. Here you see some of the key members of truly international partnerships that have enabled ivermectin to reach those in need, no matter where they are. Merck, TDR, EPOC, and of course, affected communities themselves all work well together to achieve the excellent result we are witnessing today. In 2004, I went to Africa to see the real impact that ivermectin is having. I went to as a vendor where in the 1970s, many of the adult villagers had been blinded by onchocerciasis. The blind farmer you see here are taking a Mekuchizan tablet every year. Skin itching has been dramatically reduced, although blindness cannot be reversed. However, by taking ivermectin, the infection will not be transmitted. Since 1987, some 37 million African children have been saved from the treat of onchocerciasis by ivermectin. Lymphatic filariasis also result from the infection with parasitic filarial worms transmitted by mosquitoes. Infection causes filarial fever, elephantiasis, genital damage, and social stigma. This was the position in 2000. More than 1.3 billion people are at risk. This number is almost 20% of the world population. 120 million people already infected, and this disease affected 83 countries. In the year 2000, the drug donation programs were extended to cover this disease where it coexists with onchocerciasis. Today, these numbers are dramatically reducing due primarily to ivermectin. Since the donation program began in 1987, 2.6 billion ivermectin treatments have been approved. Latest data show that around 227 million people are taking donated ivermectin annually. Today, no child in Africa will face the prospect of being blinded by onchocerciasis. The health and socio-economic welfare of millions of families in Africa and Latin America are being saved from the dangers of both liver blindness and elephantiasis, thanks to a single soil organism, concerted scientific mind, and collaborative endeavor. Within the decade, two of the world's most devastating and disfiguring diseases should soon be eliminated as public health problems: lymphatic filariasis by 2020 and onchocerciasis by 2025, or even sooner. None of these children will be treated by either disease. No child born in Africa after 1987 will face the threat of blindness or disfigurement, thanks mainly to ivermectin. That my work helped active this is a reward beyond measure. But the success does not stop there. Around the world, commercial formulation of ivermectin are also first-line drug for various diseases, including thromboidiasis and scabies. As you see here, evidence is also accumulating that giving ivermectin to entire community improves all sorts of health issues and socio-economic factors, not just overcoming onchocerciasis and lymphatic filariasis. We are currently funding a project to quantify the complete impact of long-term ivermectin consumption in communities in Nigeria. We have also donated bicycle and mobile phone to help make the job of volunteer village-based ivermectin distribution much easier. Research is also showing that ivermectin has a significant impact against a variety of neglected tropical diseases. We have set up a research collaboration with the Oswaldo Cruz Institution in Brazil to test ivermectin against a number of other neglected tropical diseases, and have already got some encouraging results with respect to schistosomiasis. I would like to indicate the Japanese community also benefited from our discovery. The royalty we earn on sale for animals have been used to fund continuing research, as well as to pay for construction of this 440-bed district hospital, nursing college, dormitory for nurses, and vaccine production facilities. Today, in addition to inpatient, over 1,000 outpatient pay per day visited the hospital. Ivermectin is without any doubt a splendid gift from the earth. Now, I'd like to talk briefly about our genetic work on the ivermectin producer to understand and exploit the way microbes produce metabolite. We undertook complete genome analysis of the organism, which, as I mentioned, is the only source for ivermectin ever found. We first reported our finding, which covered 99.5% of the total genome in 2001, and completed the analysis of entire genome in 2003. This is the issue of Nature Biotechnology, in which our paper on entire genome analysis appeared with an electron micrograph of the organism we discovered on the cover. The information obtained, which represented the first genome analysis of an industrially important actinomycete, triggers intensive research on the secondary metabolite of microorganisms worldwide. This slide shows the outline of the genome. There are more than 9 megabase, highlighting the genes cluster involved in the biosynthesis of metabolite. Italic abbreviation under each molecule structure indicated the biosynthetic genes whose location is indicated by arrow on the linear chromosome. Underlined italic abbreviation indicated that products were identified in the fermentation broth of this strain. For example, in addition to ivermectin, oligomycin, pentyl macrolide, squalene, and carotene. Fortunately and astonishingly, no ivermectin resistance has been reported in humans, despite over 30 years of monotherapy. But we are continuing to create a new analog via genetic manipulation to help offset any problem should resistance develop, as it has done in animals. The upper figure is the gene cluster for ivermectin biosynthesis. These 4 genes, they have a 1 to 4 product. Multifunctional protein that catalyze the formation of lactone skeleton. So please take a look at the elegant work of organism in ivermectin producing process. The elongation and lactone forming modification, and to this lactone, the oleandrose was synthesized from glucose and linked to the 13 position. Thanks to our genetic work, we can now control and manipulate this process, opening up the prospect of more potent and more multi-use compound. At this point, I would like to briefly explain my basic research philosophies. I should perhaps emphasize that I have already been convinced that nature holds the answer to all our needs and problems. And as you can see, my thinking has been that microorganisms are a limited resource capable of meeting our requirements. I said this was in the early 1970s. They have proved through 50 years of research. I have been lucky enough to find ivermectin and a number of other extremely useful chemicals, such as staurosporin, world's first protein kinase inhibitor of microbial origin, and lactocystin is the world's first proteasome with a microbial origin. And I shall continue to search for more and more compounds that can bring health and socio-economic benefit to communities around the world, and encourage the new generation of scientists to do the same, all in the hope of discovering a new ivermectin or yet more such a wonder drug. For 50 years, I have worked alongside specialized researchers in the fields of such as biochemistry, molecular biology, microbiology, and clinical medicine. My approach has always been influenced by the tenet, one encounter, one chance. It captures a deep respect that is an essential part of the tea ceremony, Tu Youyou. Tu Youyou is held in the highest esteem in Japanese culture, as well as the fact that exact circumstance at any point in time will never happen again. I believe it is important to seize opportunity when they appear, and to maintain profound respect and consideration for all my colleagues, as well as for the microorganisms I work with. Such a sentiment forms the basis for all good scientific research and discovery. To conclude, I would like to once again express my sincere gratitude to all concerned for being chosen as the recipient of Nobel Prize in Physiology or Medicine, and I humbly accept the award on behalf of all those who have helped me on every step of a wonderful journey of discovery. So thank you very much.
William C. Campbell received a BA from Trinity College, University of Dublin, in Ireland, 1952, and a PhD from University of Wisconsin, Madison, Wisconsin, in the United States in 1957. He was with the Merck Institute for Therapeutic Research from 1957, and in 1984, he became the Senior Scientist and Director for Assay Research and Development. Campbell is currently a Research Fellow Emeritus at Drew University, Madison, New Jersey, USA. Please join me in welcoming Dr. Campbell, who will present his Nobel lecture under the title Ivermectin, a reflection on simplicity.
Your Excellencies, ladies and gentlemen, as you see from my title, I intend to talk about simplicity. Simplicity in the context of science. And in talking about simplicity, I do not mean to suggest that science itself is simple, nor will I suggest that the discovery and development of the drug ivermectin was an exercise in simplicity. I want rather to call attention to the element of simplicity within science. And I want to do that by pointing out the prominence of simplicity in the genesis of ivermectin. It has, of course, long been acknowledged and recognized that simplicity has an intrinsic appeal to scientists as it does to others. And indeed, simplicity is widely celebrated in science as a matter of beauty. But I want to talk about simplicity not as a matter of beauty, but as a matter of practical utility. Consider a real-life, actual event. On a particular day, the 9th of May, 1975, there was a mouse in a mouse box in a laboratory. It had been purposely infected with worms, but not enough to cause illness. And on that particular day, the mouse's diet was altered. It was altered by having some liquid stirred into its regular food. And it ate that food for almost a week. And then its regular diet was restored. And almost a week after that, its worms were gone. The mouse had been cured of its worminess. And from that moment, there was a train of events set in motion that would lead to an advance in medicine, both human medicine and veterinary medicine. And that advance in the basic science would in turn lead to practical changes in the management of diseases. So to a very large extent, the drug ivermectin was brought about by simple science. It was not conventional science, it was not obvious science, but it was simple science. And I want to make a slight digression here in relation to another question. I have often been asked in recent weeks how it felt when I heard that I had won the prize. And I can say without hesitation that my mind was instantly flooded with two emotions, one an emotion of joy and gratitude, and the other emotion, one of sadness. And the sadness because I knew instantly that all the people who contributed to this drug and made it a success could not be named individually. And so I am a representative of the Merck & Company research team. And in that capacity, I am grateful and honored beyond imagining. So, what was in that liquid that was added to the regular food of that mouse on the 9th of May? It was a liquid in which a microbe, a bacterium, had been allowed to multiply, breaking down the surrounding nutrients and producing all kinds of byproducts, metabolites of various kinds. In other words, the liquid that was added to the diet had been fermented by a bacterium. And that bacterium had been isolated and sent to Merck & Company by Dr. Satoshi Ōmura, whom you have just heard, and who shares with me the prize that has brought us here together today. And many years later, I had the pleasure of visiting him in Tokyo. Microbes do not all look alike or act alike, but the Kitasato Institute and Merck, both of them, were interested in finding microbes that stand out from the crowd. Microbiologists get tired of finding microorganisms that have already been found. Professor Omura sent us microbes that were unfamiliar in general to microbiologists. And we found that one of those unfamiliar microbes produced an unfamiliar substance, and that that substance had very potent activity against worm parasites, and also against some other parasites, but that is a story for another time. The mouse that I mentioned was a single mouse. I do not mean that the mouse was unmarried. I mean only that special diet had been tested in only one mouse. Usually things are tested in groups of mice, experimental groups, but that was tested in just one single mouse. Other individual mice got other diets altered by other liquids in which other bacteria had flourished, but only one mouse, only one of those liquids ended in the cure of worms. Just get some water. Excuse me for a moment. That test method, which might strike you as unusual, was developed in the parasitology department of Merck and Company by Dr. John Egerton. And that is also where, in the same department, Dr. Dan Ostland had pioneered the reduction of experimental group size to a singleton. Simplicity in the history of ivermectin was just a beginning. From then on, there was complexity, many years of basic, complex research, and many years of complex developmental research. Pharmaceutical development is the epitome not of simplicity, but of complexity. And many scientists and many disciplines were involved in the development of this drug. And I will mention perhaps one or two, perhaps biased towards parasitological matters, but I will have, I trust, in the written version of this talk, opportunities to acknowledge my colleagues and family and friends more generously. Many things were going on at once at this time. The microbiologists described the bacterium as a new species of Streptomyces. The fermentation chemists and biologists isolated the mystery substance that killed worms, and they persuaded the microorganism to produce that substance more abundantly. And analytical chemists determined its chemical structure. It's actually a set of closely related molecules that we named ivermectin. The synthetic chemists found how to make an even better version of it, and so I will show you the structure. This is a picture I took of Dr. Omura in 1990, and you can see that he has not changed at all. This is the original stuff, ivermectin, or ivermectin B1A. This is a simplified slide. There's a lot of detail left out of this. It's also known as abamectin. So that's the original stuff, unmodified. But our organic friends in chemistry, they acknowledge that microorganisms can make molecules that humans cannot, but they also seem to believe that only a chemist can make it properly. And so they tweak these things and modify it, and so what they did was to make a better version of it, which is shown here. Now, I trust you all have noticed the difference, but just in case you haven't, just focus on the upper right-hand corner, where you see here and here, the 22-23 bond between those carbon atoms. Each of them has been has been given another hydrogen. So this was accomplished by hydrogenation of ivermectin to make ivermectin. And so the ivermectin is 22-23 dihydroivermectin B1A. B1A just being one of these closely related molecules. So they had made this change by adding hydrogen. So they thought, not surprisingly, that they would like to call this stuff not ivermectin, but hyvermectin, because hydrogen had been added. But it was pointed out to them that in some language, hyver means testicle. And so that's how ivermectin became ivermectin. Despite being unique in its origin, ivermectin now has many relatives. Many of them are on the market in animal health. And there's a whole family of these related compounds called macrocyclic lactones, and there is vast literature on those. Meanwhile, the parasitologists had found out which worms it would kill, and the biochemists found out how it would kill, or rather how it would paralyze them. Because with parasitic worms, paralyzing them is just as good, or from the worm's point of view, just as bad, as killing them. The body itself will get rid of paralyzed worms. With this flood of information that was being gathered, the project was given developmental status, so even more disciplines, more scientists became involved. And I will just mention one as an example of many very different scientific disciplines. And I want to mention for a moment the veterinary parasitologists. We had many veterinary parasitologists, not only from our own country, but from veterinary schools all over the world. And they were brought into this project. And they had an astonishing breadth and depth of knowledge of parasites. And they set up trials to assess the efficacy of ivermectin against many different kinds of parasites in many different kinds of host animals in many different lands. And I think their expertise was actually essential to the success of ivermectin in animal health, where it became the dominant antiparasitic agent. Sometimes things that one are thought of as bad turn out to be good. If a broad-spectrum antiparasitic agent fails to be active against some particular important pathogen, that is usually oblivion for that drug candidate, if it misses some very important parasite. But not necessarily so. In the case of dog heartworm, the significant pathogen is the adult worm stage, and ivermectin is not active against adult heartworm, Dirofilaria immitis, in dogs. However, in the routine treatment of dogs to get rid of their worms, and I emphasize in the routine treatment that goes on all the time, it is actually dangerous to kill that adult worm. Killing the adult worm is quite likely to end in the death of the dogs. It can be fatal to dogs. It can be fatal to the reputation of the veterinarian. And so that is not a good thing to do. So ivermectin turns out to be ineffective exactly where you would like it to be ineffective. Still, ivermectin is used all the time to control heartworm disease in dogs. It's used as a preventative agent. And as it happened before the discovery of ivermectin, I had instituted a program for heartworm research at Merck. And Linda Blair and I had set up the life cycle in the lab, mosquito to dog life cycle, and we had that going just when we needed it to test ivermectin against heartworm. And we found that it's active against a pre-adult phase, so we were able to do the research that led to the development of a once-a-month treatment, the first once-a-month treatment for the prevention of heartworm in dogs. The potential value of ivermectin in humans was never overlooked. I had always insisted that our written objectives include the development of drugs for the control of parasites of people. And when my colleagues, John Egerton and Linda Blair, found that ivermectin was active against the larvae of the parasite Onchocerca cervicalis in the skin of horses, then I knew it was time to take further action. And my chief, Dr. Jerry Birnbaum, enthusiastically approved my proposal to invite Dr. Bruce Copeman in Australia to do a trial against another onchocercal species, Onchocerca gutturosa in cattle, and we gave him funds to support that research, which he did, and he's an expert on it, and again he found that ivermectin was effective. So at that time, we had information suggesting that ivermectin might be useful in humans against a variety of parasitic worms, including hookworm, for example. But it was clear when one would evaluate the situation that the best way to meet an unmet need in human medicine would be to have it used against river blindness, which is caused by yet another species of Onchocerca, Onchocerca volvulus. So Dr. Birnbaum and I took that message to the top level of research management at Merck and Company. It was an exciting moment for both of us. We made our pitch, and at that time, the head of research at Merck was Dr. Roy Vagelos, who himself was a physician and a chemist. And they listened very intently to our story, and they approved a trial of ivermectin in humans, a very cautious trial in patients that had the very early stages of river blindness, long before there was any actual blindness. They would have the infection that causes river blindness, but they would have light early infections. So this was a very cautious trial at the beginning, and this was one of a series of trials that was set up and directed by Dr. Mohammed Aziz, who was one of Merck's clinical directors, and he was in charge of doing this. And he set up this trial in Senegal, and it was undertaken by Dr. Aziz and Dr. Diallo of the University of Dakar and Dr. Lariviere of the University of Paris and their colleagues. And they showed that ivermectin was active against the microscopic larvae, the baby worms in the skin of people. And that was, of course, turned out to be exactly what is needed. The trial had to be very cautious because there had been some precedent that it was dangerous to kill those microscopic worms, but this turned out not to be dangerous and, in fact, to be very safe. And to understand why the activity of that against those larvae is important, it's necessary to understand that in river blindness, unlike the situation with dog heartworm, it is the offspring, these microscopic progeny, that cause the damage to the skin and to the eye, and eventually leads to blindness, as well as skin disease and serious itching. And so killing those larvae can prevent the onset of disease. You can block it before it gets going, and that's what ivermectin does. In 1987, Dr. Vagelos was then CEO, president of Merck and Company, and he announced that the company would donate the drug for the use against river blindness. And this led to an unprecedented donation of the an unprecedented translation of this donation of the drug into the actual distribution of the drug. And that was a very complicated business, and it was undertaken by many groups. I will just mention a couple of them. You've heard of some of them already. But I mentioned the World Health Organization and Merck's own Mectizan donation program, which was set up from the very beginning to make sure that it was properly used, and the Carter Center in Atlanta, Georgia. But there were many, many other agencies involved in this process. And also there were many leaders involved in this process, many directors of the various components. And I will just mention one of them. And I feel I must mention one because he is Swedish. He is Dr. Björn Tillefors, and I am honored that he is here for the activities of this week. I will now show just a few more slides. This is a sculptural representation of a well-known, iconic scene in endemic areas, where you have endemic areas that are endemic for river blindness, where you have adult people who are blind being led by children and often connected with a stick, sometimes 3 or 4 or 5 people all connected to one child. And this is a well-known thing, and this is where the river blindness program comes in. And the impact of ivermectin and river blindness in terms of the extent of that impact, which has now been developing over many years, is something that is now well documented and well established, and not for me to discuss here. I just want to show a few pictures of some of the very early treatments that I was an observer for in the early program. This is Dr. Mohammed Aziz, the Merck clinical director, who was responsible and who chose the place to do the first trial, chose the place to people to do it with, and made sure that all was done according to proper protocol. I'll show another slide of people. There's 3 people here of great significance. My wife and I are talking to 2 of the people. We're on the left, and I don't need to say why my wife is important. You already know that. But the 2 people on the right are of tremendous importance. One is Jimmy Carter, the head of the Carter Center, former president of the United States. And he, because of his past position, was in a position to be very influential in helping the distribution program. And then standing next to him, second from the left, second from the right, in this picture is Dr. Roy Vagelos. And he was the one who had to make the decision what to do with this drug, and he was the one who decided it would be donated. And there is no point really in talking about various people who have suggested it in various parts of the world at various times, because the important thing is who considered it, who were in a position to do something about it. If you have to be accountable for it, then it becomes a big decision to make, and that's why Roy Vagelos made such an important decision. Now, I would like to not talk further about how great an impact this was, but rather I want to get back to that mouse assay, the test system with which I began. The operation of that assay was simple, as I have explained. The operation was simple, but so was the principle of it. Thinking it up was a different matter. That was innovative thinking on the part of my colleagues. But the underlying principle was simple. It was bizarre, but it was simple. And I have described it this way. You line up a series of infected mice. You treat each mouse with an unknown amount of an unknown substance that might not be there. And then you check to see if it worked. On the face of it, it seems to fly in the face of what we are taught about science with its well-regulated system and its emphasis on measurement. But we need to understand that this, too, is well-regulated science. Empirical research based on observation and trial and error and screening of substances has been the foundation of the discovery of antiparasitic drugs. But in recent decades, that screening approach has been beaten into disrepute and desuetude, and has in many places been abandoned. And when it is abandoned, we cannot know what price has been paid in non-discovery. I have recently made a proposal. Before I get to that proposal, let me just show these last few slides. I had a chance to observe the early trials. This is in a remote village in northern Togo, where I saw the first community-based trials in remote places like this, where villagers would gather together in a schoolyard under the tree for treatment. You see on the left a girl standing on a bathroom scale to be measured, because at this stage very detailed records were kept. It turned out later that that bathroom scale was an unnecessary expenditure of high-tech instrumentation, and it was long since replaced by a measuring stick, which is much easier to use and much more efficient. Children were given a tiny pill, which they could easily swallow and have a drink of water and open their mouths to show that they had swallowed it. This is a village elder. When you, in one of these gathering situations where the treatment was being given, when you would see people like this, a blind elder who had the leopard skin caused over many, many years of infection with river blindness and had been preceded by really excruciating itching. Unfortunately, in those situations, you would also see a man like this, a blind man for whom treatment had come too late, because ivermectin will prevent blindness, it will not cure it. But what the good part of those collections of visitors under the tree or in a schoolyard was seeing collections of children who would now be safe and would no longer grow up under the threat of the skin disease and the itching and the eventual blindness. I recently made a proposal for more broadly searching the world for natural products, especially microorganisms, as a means of finding new molecules for development as chemotherapeutic agents. And that might be useful to supplement to some of the mass treatment programs that are currently underway to treat various diseases. But nevertheless, chemotherapeutic control should not be seen as the ultimate objective. Unnatural measures are likely to have unforeseen natural consequences. And the broader the activity spectrum of a biodynamic substance, the more we must guard against the hazards of indiscriminate use. And since we cannot count on discriminate use, we should try to control the disease without recourse to chemical agents. There are very few acceptable anti-parasite vaccines, but we might yet learn how to make better ones and more of them. And we also might be able to devise public health measures to subvert parasite life cycles more subtly than in the past. In late afternoon, I often climb a nearby hill. Not a mountain, it's just a grassy half-mile hill called Half Mile Hill. And from the top, there is a marvelous vista of woodland and lake, and the sky often tinted with color as evening falls. And it is a moment of uplifting tranquility. But one realizes, of course, that not everybody lives amidst such natural beauty and in conditions of peace. And around the world, there are many people making heroic efforts to redress that terrible imbalance. And one of their objectives is to improve global health. And as we bring science to bear on such problems, I hope we will keep in mind that solutions are sometimes to be found in science that is simple. Thank you.
Tu Youyou graduated from the pharmacy department at Beijing Medical University in 1955. She was appointed assistant professor at the China Academy of Traditional Chinese Medicine in Beijing in 1965 and became associate professor in 1979. She was appointed professor at the same institute in 1985. From the year 2000, Tu has been chief professor at the China Academy of Traditional Chinese Medicine. Please join me in welcoming Dr. Tu, who will present her Nobel lecture, Artemisinin, a gift from traditional Chinese medicine to the world. She will present in Mandarin with translation into English.
席先生，尊敬的获奖者、女士们、先生们，今天我极为荣幸能在卡罗林斯卡学院演讲。我报告的题目是青蒿素，中医药给世界的一份礼物。在报告之前，我首先要谢谢诺贝尔奖评委会、诺贝尔奖基金会授予我2015年生理学和医学奖。这不仅是授予我个人的荣誉，也是对全体中国科学家团队的嘉奖和鼓励。在短短的几天里，我深深地感受到了瑞典人民的热情，在此我一并表示感谢。谢谢刚才两位所做的精彩报告。我现在要说的是，40年前，在艰苦的环境中，中国科学家努力奋斗，从中医药中寻找抗疟新药的故事。关于青蒿素的发现过程，大家可能已经在很多报道中看到过。这里我只做一个概要的介绍。这是中医研究院抗疟药研究团队当年的简要工作总结，其中蓝底表示的是本院团队完成的工作，白底表示的是全国其他协作团队的完成工作。蓝底向白底过渡，表示既有本院也有协作单位参加的工作。中医药研究所团队于1969年开始抗疟中药研究，经过大量的反复筛选工作后，1971年起工作重点集中在中药青蒿。又经过很多次失败以后，1971年9月重新设计了提取方法，改用低温提取，用1米回流或者冷沁，然后呢，再用解溶液去除酸性部位的方法制备样品。1971年10月4号，青蒿1米中性提取物就是标示的191号样品，以1公斤体重1克的剂量，连续3天口服给药，取疟药效评价显示抑制力达到100%。同年12月到次年1月，猴疟实验也得到了抑制力100%的结果。青蒿1米中性提取物抗疟药效的突破，是发现青蒿素的关键。1972年8月到10月，我们开展了青蒿1米中性提取物的临床研究，经过30例恶性疟和间质疟病人治疗全部见效。同年11月，就从这个中性部位中成功的分离得到抗疟有效单体化合物的基金，就是后来命名为青蒿素的基金。1972年11月开始，对青蒿素的化学结构进行探索，通过元素分析、光谱测定、质谱循环分析等技术手段，确定化物分子式是碳15氢22氧5，分子量282，明确了青蒿素是不含氮的倍苯萜类化合物。1973年4月，经中国医学科学院药物研究所化学分析时，进一步复核了分子式等有效数据。1974年起，与中国科学院、上海有机化学研究所和生物物理所相继开展了青蒿素结构协作研究的工作，最终经 X 光衍射确定了青蒿素的立体结构， 确认青蒿素是含有过氧集团的新型倍苯天类子。这个结构在1977年在中国的科学通报上发表，后来就被 CA 所收录。1973年起，为研究青蒿素结构中功能集团而制备的衍生物，经硼氢化钠还原证实青蒿素结构中羰基的存在，发明了双氢青蒿素。经高效关系研究，明确青蒿素结构中的过氧集团是抗疟的活性集团。部分双氢青蒿素的羟基衍生物的除疟效价也有所提高。这里展示了青蒿素基衍生物双氢青蒿素、蒿甲醚、蒿乙醚、青蒿糊酯的分子结构。直到现在，除这一类型之外，其他结构类型的青蒿素衍生物还没有用于临床的报道。1986年，青蒿素获得了卫生部新药证书。1992年，再获得双氢青蒿素的新药证书。双氢青蒿素的药效高于青蒿素10倍。进一步体现了青蒿素类药物高效速效低毒的特点。青蒿素引起世界关注。1981年，世界卫生组织、世界银行、联合国计划开发署在北京联合召开了疟疾化疗科学工作组第4次会议，有关青蒿素及其临床应用的一系列报告在会议上引起了热烈的反响。我的报告是青蒿素的化学研究。上世纪80年代，中国已经有数千例的疟疾患者得到了青蒿素及其衍生物的有效治疗。听完这段介绍，大家可能会觉得这不过是一段普通的药物发现过程，但是当年在中国已有2000多年研用历史的中药青蒿中，发掘出青蒿素的历程却相当艰辛。目标明确，坚持信念是成功的前提。1969年，中医科学院中药研究所参加全国523抗疟研究项目，经院领导研究决定，我被指令负责，并组建523项目课题组，承担抗疟中药的研发。这个项目在当时属于保密的重点军工项目。对于一个年轻科研人员有机会接受如此重任，我体会到了国家对我的信任，深感责任重大，任务艰巨，但我决心不辱使命，努力拼搏，请全力完成任务。学科调查为研究发现成功提供了准备。这是我刚到中药研究所的照片，左侧是知名生物学家娄志勤教授指导我鉴别药材。从1959年到1962年，我参加了西医学习中医班，系统学习了中医药的知识。化学家卢耶普斯特说过：“机会垂青有准备的人。”古语说：“凡是过去，皆为续缺。”然而，续缺就是一种准备。当抗疟项目给我机遇的时候，西学中的续缺为我从事青蒿素研究提供了良好的准备。信息收集为准确解释是可研究发现成功的基础。接受任务以后，我收集整理历代中医药的典籍， 走访名老中医，并收集他们用于治疗疟疾的方剂和中药，同时调阅了大量的民间方药，在汇集了包括植物、动物、矿物等2000余内服外用方药的基础上，编写了以640种中药为主的疟疾单列方集。正是这些信息的收集和解释，铸就了青蒿素发现的基础，也是中药新药研究有别于一般植物药研究的地方。关键的文献显示，当年我面临研究困境时，又重新温习中医古籍，进一步思考到东晋葛洪走后北极方有关青蒿一物以遂两心则交取之进服之的经验记载，这使我联想到提取过程可能需要避免高温，由此改用低伏点用具的提取方法。关于青蒿入药，中国最早见于马王堆3号汉末的帛书《52病方》，其后的《神农本草经》、《溥仪雷公炮制编览》、《本草纲目》等典籍都有青蒿治病的记载。然而，古籍虽多，却都没有明确表明青蒿的植物分类品种。当年青蒿资源比较混乱，药典就收了2个品种，还有4个其他混交品种也在使用。后续研究发现，经 Artemisia annua 一种还有青蒿素对抗疟有效，这样客观上就增加了发现青蒿素的难度。再加上青蒿素的原始物种含量高，还有药用部位、产地、采收季节、纯化工艺等的影响，所以青蒿素中性提取物的成功确实来之不易。中国传统中医药是一个丰富的宝藏，值得我们多加思考、发掘、提高。在团队面前，需要坚持不懈。70年代，中国的科研条件比较差，为供应足够的青蒿有效部位用于临床，我们曾用水缸作为提取用剂。由于缺乏通风设备，又接触大量溶剂，导致一些科研人员的健康受到了影响。为了尽快上临床，在动物安全性评价的基础上，我和科研团队成员自身服用有效部位提取物，以确保临床病人的安全。到青蒿素片剂临床使用效果不理想的时候，经过努力坚持，深入探究原因，最终查明是泵解度出的问题，改用青蒿素代替胶囊，从而及时地证实了青蒿素的抗疟疗效。团队精神无私合作，加速科研科学发现转化成为有效药物。1972年3月8日，全国53办公室在南京召开抗疟药专业会议，我代表中药所在会上报告了青蒿素191号的提取物对畜疟由猴疟的100%结果，受到会议极大的关注。同年11月17日，在北京召开的全国会议上，我报告了30例临床全部有效的结果，从此拉开了青蒿素抗疟研究全国大杰作的序幕。今天我再次衷心感谢当年从事523抗疟研究的中医科学院团队全体成员，铭记他们在青蒿素研究、发现与应用中的积极投入与突出贡献。 感谢全国完善项目单位的通力协作，包括山东省中药研究所、云南省药物研究所、中国科学院生物物理所、中国科学院上海有机所、广州中医药大学以及军事医学院、医学科学院等等。我衷心祝贺协作单位同行们所取得的多方面成果，以及对疟疾患者的热忱服务。对于全国完善办公室在组织抗疟项目中的不懈努力，在此表示诚挚的敬意。没有大家无私合作的团队精神，我们不可能在短期内将青蒿素贡献给世界。疟疾对世界公共卫生依然是个严重挑战。WHO 总干事陈凤富珍在谈到控制疟疾时，有过这样的评价：在减少疟疾病例与死亡方面，全球范围正在取得的成绩，给我们留下了深刻的印象。虽然如此，据统计，全球97个国家和地区的33亿人口仍然遭遇疟疾的威胁，其中12亿人生活在高危区域，这些区域的患病率有可能高于1/1000。统计数据表明，2013年全球疟疾患者约为1.98亿，疟疾导致的死亡人数约为58万，其中78%是5岁以下的儿童，90%的疟疾死亡病例发生在重灾区非洲。70%的非洲疟疾患者应用青蒿素的复方药物治疗，但是得不到 ACT 治疗的疟疾患儿仍能达到5600万到6900万之多。疟原虫对青蒿素和其他抗疟药的抗药性，在大湄公河地区，包括吉普赛、老挝、缅甸、泰国和越南，恶性疟原虫已经出现对于青蒿素的抗药性。在吉普赛、泰国边境的许多地区，恶性疟原虫已经对绝大多数抗疟药产生抗药性。请看今年报告的对于青蒿素抗药性的分布图，图上红色的、黑色的，提示当地恶性疟原虫出现抗药性。可见，不仅在大湄公河流域有抗药性，在非洲少数地区也出现了抗药性。这些情况都是严重的警示。世界卫生组织2011年遏制青蒿素抗药性的全球计划。这项计划出台的目的是保护 ACT 对于恶性疟疾的有效性。鉴于青蒿素的抗药性已在大湄公河流域得到证实，扩散的潜在威胁也正在考察之中。参与该计划的100多位专家认为，在青蒿素抗药性传播到高感染地区之前，遏制和消除抗药性的机会其实十分有限。遏制青蒿素抗药性的任务迫在眉睫。为保护 ACT 对于恶性疟疾的有效性，我诚挚地希望全球抗疟工作者认真执行 WHO 遏制青蒿素抗药性的全球计划。在结束之前，我想再谈一点中医药。中国医药学是一个伟大的宝库，应当努力发掘，加以提高。青蒿素正是从这一宝库中发掘出来的。通过抗疟药青蒿素的研究历程， 我深深地感到中西医药各有所长，两者有机结合，优势互补，但具有更大的开发潜力和良好的发展前景。大自然给我们提供了大量的植物资源，医药学研究者可以从中开发新药。中医药从神农从百草开始，在几千年的发掘中积累了大量的临床经验，对于自然资源的药用价值已经有所整理归纳。通过继承发扬、发掘提高，一定会有所发现，有所创新，从而造福人类。最后，我想与各位分享一首我国唐代有名的诗篇，黄之欢所写的《登巨阙楼》。白日依山尽，黄河入海流。欲穷千里目，更上一层楼。请各位有机会时更上一层楼，去领略中国文化的魅力，发现永恒于传统中医中的宝藏。衷心感谢在青蒿素发现、研究和应用中做出贡献的所有国内外同事们、同行们和朋友们。深深感谢家人一直以来的理解和支持，衷心感谢各位前来参会，谢谢大家。
The 2015 Nobel Lectures in Physiology or Medicine have now come to an end. I would like to extend the heartfelt gratitude of Karolinska Institutet to the laureates for their spellbinding presentations this afternoon, and wish Tu Youyou, William Campbell, and Satoshi Ōmura some memorable days to come. Thanks also to you in the audience for sharing this significant moment with us. May I now ask Doctors Campbell and Omura to join us here on stage for the compulsory photo session. And also, may I call upon the head usher to provide some housekeeping information.
Thank you all for attending the Nobel Lectures in Medicine or Physiology. And here's some practical information. The members of the Nobel Assembly and the guests of the Nobel laureates, please follow our ushers up the door. who will guide you, and all the other guests, please exit through the side doors to the left and to the right, and down the stairs to outside. So thank you very much.